important. Now, when we have this agar comes out, why it is very magic stuff? Agar can be melted at 50 degrees Celsius. And we will do this in the lab. You will see. It will be melted at 50 degrees Celsius. It will be solidified very well, averagely evenly at 25, 30 degrees Celsius. And we could do a streak plating, spread plating, and pour plating to put the bacteria there, and the bacteria can grow very well forming colony. And we will know those are the pure culture. Okay? So how are we going to do that? This is what we're going to practice in the lab. This is the agar. How are we going to get the pure culture? Let's say it's a bacterial broth. We were using a loop to get a little bit at the beginning. Very tiny. Evenly spread. Then we're going to flame the loop to the second part. Less intensive. Then we flame the loop again from the side to do a third course. More or less intensive than the last one. Will be like that. Then this agar we put at 35 degrees Celsius, incubate 24 hours. Some dramatic stuff will come out. These single dots will be surrounded by the line what you streaked. This is a pure culture. So what is a pure culture? It is a group of bacterial cells coming from the same mother cell. And this pure culture, we also call it a colony. This one dot, we call it a colony forming unit, CFU, the acronym. But here is something I will also want to tell you. This one colony does not mean one cell. So do not be confusing by yourself. How many cells are there? At least 10 to the 6th to 10 to the 8th cells there. That's one colony. But they're all coming from the same mother, so it's a pure culture. Now, why this pure culture is so important? Because later on, we want to know the genetic information. We want to know the biochemistry characteristics. They all need to be from the same group. If you messed up with other group, the results will be complicated and very difficult to explain. So that's why this pure culture is very important. Now, last step, we have the agar. We need a container. So, in the old time, people using a rectangle shape as a container. And some people think about we're going to using as a round circle as a container. It's a simple mesh, same perimeters. The circle has a larger area. So they need using the container to hold on the agar, which is a circle. Who generated that? The name called Richard Petrie. That's why right now, all the agar, when we pour them, let them be solidified, the container, we call it Petri dish. Because it's come out from Richard Petrie, another great scientist from Bob Koch School. Okay, so this is pretty much the story of isolation of pure culture. So we can see here, the pure culture from the agar, it's a polysaccharide isolate from seaweed, and the bacteria can grow it. solidified at the room temperature, melted at a high temperature. This is a strict plating methods. We will practice a lot in the lab. And I just draw the picture here to explain to you. And I also, we call this is a serial dilution on the solid agar medium. You do this a lot in your biochemistry chemistry class, which is a serial dilution in cubes. And we don't do this often, is a serial dilution on the solid agar medium. But we will do this in the lab, okay? So that's, you can see the colonies there. The colony indicate a few cultures of the bacteria. Last thing, we still want to talk about the binomial system of a bacteria, which is basically a name system of the bacteria. There is a lot we can talk. Something important. People doesn't realize that too often. We want to emphasize here in the first day of the class. Okay? So first of all, how we name a bacteria? It's called a binomial nomenclature. Binomial name system, let's say. So how do you do it? The first one, let's say E. coli, is Escherichia coli. Escherichia is genus name. Coli is a species name. You have to do it italic. That's important. Escherichia coli, the Escherichia is a scientist name from German, a Japanese scientist name. He's the first person finding E. coli in the fecal samples. And the coli, COI, is usually the shape. It's a large shape. So that's why we say COI. And Listeria monocytogenes, for example. Listeria that's come from a person, a monocytogenes, which means it's a single cell unit. Okay. Let's say strep throat, streptococcus, COCCUS pyogenes. Streptococcus is a shape, pyogenes, pus and genetic. Okay, you have to italic. That is called a binomial system. But there is an exception. The exception is a pathogen everybody heard most of the time but didn't realize is Salmonella. This is the exception. The correct way to write a Salmonella, usually the first part is italic, and the second part is normal. Let's say Salmonella Newport. You should write it like that. But why? It's a very good question for the microbiologist all the time. Because Salmonella Newport is supposed to be a very long name. Is Salmonella enterica subspecies enterica Newport. From the beginning to here, it is all italic. The last one is normal. Because it's a very long subspecies name, lots of the time people skip this part. If you connect these two, it becomes this. So what is the reason Salmonella Newport is not italic? Because this is not a species name. This is subspecies name. And the E. coli here is a species name. Okay, that's some tricky behind that. Lots of people doesn't know. If you read a paper, you read a book, you see Salmonella typhimurians, Salmonella Newport, Thompson, Hindenburg, all those, they both write italic. That means the people doesn't understand the microbiology very well, because that's a tricky concept behind that. Last thing I want to mention, we talk a little bit about in our first class. We want to give you some of the example, which is the E. coli. And I made a joke, I said, E. coli you're going to remember. Even if you failed this class, and many years later you still remember the only bacteria name in this classroom is E. coli. So we want to talk a little bit about that. When you see E. coli, Escherichia coli, looks like a genus name, species name is very simple, and you think it's bad, but I tell you it's not necessary. First of all, there is a generic E. coli. Generic E. coli, usually we call it an indicator bacteria. That's a bacteria to indicator of fecal contamination. So if in the pond water, let's say, or river, or some of the lake, you get pond water, so you isolated bacteria, you find generic E. coli. Most likely this water is contaminated with human, animal, fecal, or poop sample. So that's generic E. coli. It's an indicator bacteria. So lots of people are going to say it's a pathogen, is that right? Yes, it's a pathogen. What is that? That is E. coli, 0157H7. Now, why 0157H7 is a pathogen? Because the pathogenic E. coli have four groups. EHEC. Entero, hemorrhagic E. coli. EPEC. Enteropathogenic E. coli. ETEC. Enterotoxigenic E. coli. And EIEC. That is entero-invasive E. coli. And E. coli 0157H7 belonging to enterohemorrhagic E. coli, it will cause HUS, hemorrhagic uremic syndrome, which is blood kidney with water diarrhea, and people are going to die, especially undercooked beef patty samples, hamburger. That's why E. coli 0157H7 is zero tolerance in beef. I just say beefs in the United States. You're not allowed to have even one single cell. That's really bad. But we will talk about later what is 0157, what is H7, those means later on. Okay. Just want to let you know it's a pathogen, really bad. Another thing. Recently, people find not only this pathogenic E. coli is really horrible, there is something coming from, we call it the EXP E. coli. It's a urological E. coli. Because E. coli is the #1 pathogen cause urinary tract infection, called UTI. We will do this in the lab, later on lab 17. But I also want to tell you, E. coli sometimes is good. E. coli 9037, used for the fermentation. It's a fermented beneficial bacteria. So here is just as one example to tell you, even a bacteria have the same genus name and species name. It does not mean it's always bad. And when they have a different serological typing, it could be an indicator bacteria, which is a normal bacteria, could be a pathogenic bacteria, it could be a beneficial bacteria. So just want to let you know there is a lot of the magic behind that and a lot of the story behind it. It's very interesting, okay? So here we end up with our first section, which is we talk about the history and the basic information of the microbiology. And this Thursday, we will talk about mainly for microscope, okay?